There's a lot of stuff so far. Okay. So before we get started on this, I have a general question for you all. There any big problems that would come up if we moved the exam to next Friday instead of Wednesday?
Please do it.
Okay, great. So no other conflicting exams or emergencies? Awesome. Okay, then we're gonna move this exam back a day. So we'll move it to next Friday, give us a little more breathing room, a little more time to review, a little more time to let this all settle in our minds. So I'll switch that on the calendar after class, but make a note for yourselves. Awesome. All right. I'm happy about that. Sounds like you're happy about that. Glad to hear it. Okay, so today we're going to continue with our discussion of muscles. And our goal now is to think about the molecular sort of basis for how our muscles are contracting. So a muscle contraction is a shortening of muscle. And we have covered so far that the way we're shortening the muscle is shortening our sarcomeres, right? So we have our myofibril, which is our contractile sort of thread inside our muscle cell, inside our muscle fiber. We have our actin and our myosin making up our sarcomeres. So we got a Z line at each end. Right? Go ahead and draw some sarcomeres with me. So here's our actin, our thin filament. We've seen our myosin, our thick filament, center, also connected. M line. Right? We have this sarcomere, and in order to make our muscle overall shorter, we need to make this sarcomere shorter. We need to shrink it. So that's how we're going to shrink the muscle, how we're going to pull on either end. So conceptually, what we're doing is sliding the actin and the myosin past each other. Which is why we call this the sliding filament model, because our actin is the thin filament, the myosin is the thick filament. We're sliding them past each other, and as we create greater overlap between these two proteins, we're going to have a smaller overall sarcomere. So as we talk today, we're going to see these two proteins binding to each other and pulling on each other in the cross-bridge cycle in order to have this shortening effect. It's very important to remember that neither these thick nor these thin filaments, so neither the actin or the myosin is actually shrinking. We're just changing how much they overlap. So if we think about our zones, back to our sort of labeled structure of the sarcomere, we would see under a microscope side that some of the bands and zones are going to stay the same length, and some of them are going to shorten, right? So we saw that our A band, right, the A band is just anywhere we see our myosin, right? Anywhere we see that thick filament is the A band. So as these filaments slide past each other, the myosin isn't changing how big it is, isn't changing how long it is. So the A band is always going to stay the same length if we were to measure end to end. And there are regions of overlap that are changing because we have these filaments sliding past each other. So we're going to see a couple of regions shorten. So our I band here, our I band, is the region where we had actin but no myosin. So as these filaments slide past each other, as the sarcomere shortens, we're seeing this distance, right, say from here to here, it get a little shorter, not necessarily super obvious in this image, but we're changing how much overlap there is. We're having more overlap between the myosin and the actin, meaning that this region of no overlap, the I band, is shorter, smaller. The H zone was the very center here. So it encompasses mainly what we call the bare zone of the myosin here. So the part where we don't have those little heads sticking off. But it's defined as the region where we have myosin but no actin. So as we slide these past each other, we're having more overlap here in the I band region, but we're also having more overlap in that H zone region. So the H zone is also shortened. So we have two places where we have these regions shortened. And the overall effect is that the sarcomere as a whole has shortened because of this increase in overlap. Now, in order to get this sliding to happen, we have to use energy and we have to bind these two proteins to each other so they can actually tug and pull on each other. So the way we do that is through the cyclical formation of these things called cross bridges. We're going to look at the cross bridge cycle. By cyclical here, we just mean it happens over and over again. So we're going to be looking at how it happens with like one specific cross bridge, so like one head of the myosin. But a thing to realize after we learn how it goes with one cycle is that this is happening at many places across the sarcomere, right? So we have many myosin heads sticking out of the thick filament. So this is happening all over the place, and this happens kind of continuously. Each of these heads goes through, I think, like 5 cross bridge cycles a second or something. So we have more movement than is happening in just one cycle that we're about to look at. But they're all doing kind of the same thing, just slightly out of place. So here we see the sarcomere, what it would look like under a slide in these different states, in the longer state and the shorter state. So here we can see how the I band, that region where we had only actin, is shortened. That H zone, where we had only myosin, shortened. A band overall has stayed the same. And that's how they kind of figured out how this system works. So when we're generating force, we're using ATP as energy to create these links between actin and myosin, and to have the myosin actually change shape a little bit to push itself along the axis. When we talk about this, your book likes to make an analogy to an oar rowing through the water. And we'll see that that is kind of based on the shape of the myosin. We're kind of paddling towards the center, right? So if you think, I don't know if I can draw it. Let's see. So if you think about our myosin, right, as being kind of like this. So those are going to be my heads, and here I have my actin side. Okay. So we're going to be taking these oars here, these little heads, and as we push them towards the center, so as they kind of scoop like this and move that way into a new position, they're pulling the actin towards the center and creating more overlap. So that's why we think about them as like an oar paddle. So as we link together the thick filament and the thin filament, we think about that as touching our oar to the water when the two touch each other. Then we're going to have a step that we call the power stroke. That's when the myosin head is physically moving and propelling that thin filament, that actin, towards the center. Then when they detach, we pull our oar out of the water. We return to the initial position, ready to stroke through again, starting a new cross-bridge cycle. We're going to look at the molecules themselves, but this is why it makes that analogy. So the whole cycle is going to look like this, but we're going to go through it step by step. So the first thing we're going to happen, and actually I'm going to look at the full picture once before we start going into this. What I want you to realize is that right here, when we enter the cross-bridge cycle, we are in the high energy form. So we're going to be starting out with energy to push, and then we're going to use up the energy and grab some more energy at the end. So as we start the cross-bridge cycle, we have already used some ATP to get us set for this cross-bridge cycle to begin. So the first thing we have happen, so here's our myosin in this pinkish orange color, right? So here's our myosin. Here's its head, and we see what we saw as our ATPase site before. we can see that we have ADP, which is our less energized form, right, of that modified nucleotide. We can see that we bind on to those binding sites on the actin. Myosin. This one's our actin. What other molecules do we see on this image? Let's see a couple more. That's what we're labeling. What are these pink threads? Yep, this is the tropomyosin. What's going on with this sort of pink circle thing? What's that? Yeah, it's a complex. Also starts with a T. Yep, this is our troponin. And any guesses what that green thing sitting on top is? Yeah, that's our calcium. Okay. So as we're entering the cross-bridge cycle, so the reason we have this ADP and the pi bound instead of ATP is we've already broken that bond to get the energy out. So we're starting with the myosin in the high energy state. The other thing that we have already happened before this is that we had calcium enter our cell in response to an impulse coming down a neuron, activating our muscle cells, telling it to contract. So that calcium has bound to the troponin, pushing the tropomyosin aside. So all of these things have to happen first to set us up to do the cross-bridge cycle to get ready to contract. So for example, if we had the tropomyosin covering these binding sites because the troponin didn't have a calcium on it, we wouldn't be able to do the cross-bridge cycle. It's all dependent on these binding sites being available for the cross-bridge cycle. So our first thing that happens, because we have calcium, because these molecules are in a position where they can bind to each other, and because we're in this high energy form, we have the myosin bind onto the actin. So the actin binding site on the myosin is bound to the myosin binding site on the actin. The next thing that happens is we release this little phosphate ion from this site on the head of the myosin. And that leads us into our first kind of movement that we see. So as that phosphate ion detaches and falls away, because proteins work by changing their shape when they're in contact with different molecules, releasing that phosphate ion changes the shape of the myosin head. And it's gone from this position where we have like not very much of an angle at the end to a position where we've moved a little forward, right? Like we made this angle a little sharper. It's kind of subtle in the picture, but we've pushed that head a little bit forward. So that's why the arrows are all going this way, is because we are already bound on to the actin before we changed the shape. This shoved the whole molecule towards the center, towards the M line, towards the middle of the sarcomere as this changes shape. So that's the OR.
So the phosphate changes the shape of the myosin?
Yeah, as it unbinds, the myosin changes shape. Yep. So this second step is called the power stroke because that's where the actual movement is happening. So that's where the physical shortening is happening. It's already happening in step 2. But you notice that we had 5 steps to this process. And the rest of those 5 steps are going to be setting us up to do this all again, basically. In our third step, we release the ADP molecule, which is our sort of de-energized form, right? So we're releasing the ADP. And then we're going to enter something called rigor. So you may have heard the phrase rigor mortis, right? Like when cadaver bodies are all kind of stiff, right? This is actually related to that state. Rigor mortis occurs when all these cross-bridge cycles are stuck in this rigor state. This is when we've removed all of our molecules from that ATPase binding site here. We're kind of stuck in this position until we grab another ATP molecule and use more energy to put us back into our high energy state. So in death, this happens for two reasons. One, right, like you don't make more ATP because your cellular function isn't working. There's also a lot of release of calcium as the cells break down. So that's our rigor state. So first we bound the myosin to the actin. Then we did the power stroke, pushing the actin towards the center. And now we're in rigor. We're in a low energy form. We're kind of stuck until we get some more energy. So after rigor, because we are alive, we are contracting our muscle, we're going to bind new ATP to the binding site on the myosin head. Okay. So we can see that ATP bound here at that same spot where we originally had our ADP and our phosphate ion bound. So that's coming from the same spot. It's binding on. And as the ATP binds on, as we get new energy here, the myosin and the actin are detaching from each other. Right? So here we see that there's now a space. That little oar, that little foot thing isn't touching the actin anymore, even though those binding sites are exposed at this moment. Right? So the ATP has sort of triggered this shape change at this point, unbinding the two. So what happens next is ATP gets hydrolyzed. So all that means is that we're breaking the ATP down, right? So we're releasing the energy in that final phosphate to phosphate bond of the adenosine triphosphate. So we go back to the high energy form, and we can see now that broken bond. We have the phosphate ion and the ADP again, separate from each other. So now we have sort of put the energy from that bond in the ATP into the myosin molecule, put it into this shape where it's ready to grab the actin again and ready to push it through again. I'm going to scroll all the way back, and I want you guys to take a second and try and draw what you can of this from memory, just the steps. You don't have to draw all the specific stuff. And this is going to help you find where your questions are for this process. Since we're pushing the exam back a day, we can take it a little slower, which is great. How are you doing on this? see if I can draw it as we go. So what's our second step here? Yeah, 2, we got our power stroke. And why is the power stroke happening? The phosphate ion here. So we had our phosphate released. And the myosin head pulls on the axon as it changes shape. It's just a step of the cross-bridge cycle. I can't tell you more than that, unfortunately. Okay. So what's step 3? Yeah, great. Okay, rigor. I just stuck. So what's happening during 4? Great. Unbind. Axon and myosin. And what else is happening here? We bound on the ATP. And so it happens in step 5. That's the hydrolyzing part, right? So it hydrolyzed here. So now we're in the high energy state. Yeah, step 5 is the return to this high energy state. Yeah. Cool. So ATP hydrolyzing is a spontaneous process, technically. Usually doesn't happen all that fast, but it's kind of like with enzymes, how we can put molecules into specific configurations that make reactions more likely to happen. The binding to this myosin site. I haven't looked at the specific molecules, but I suspect it kind of stretches and exposes the appropriate spot. Yeah, lots of good chemistry questions. Unfortunately, I'm not a chemist. Do we like the cross-bridge cycle? Awesome. This is a major concept for this. I will try to pull out exactly what our major concepts for this exam are. I'm thinking I will give you the list of like the top 3 most important concepts when I think exactly what they are. But this is definitely going to be one of them, that cross-bridge cycle. We don't actually have a whiteboard, so we're going to skip this exercise for now, but maybe I'll bring us some stuff for next time. Okay. So next we're going to think about the excitation-contraction coupling. So all this means, excitation, here we're talking about the fact that the nerves are coming in and giving their signals to the muscle. And then the contraction part, that's just a shortening of the sarcomere using that cross-bridge cycle. Okay, so all we're talking about when we say that there's excitation-contraction coupling is that a nerve comes and tells this muscle to contract. So we're going to have a sequence of events where the action potential comes down one of those somatic neurons that we talked about way back when we were talking about the arrangement of the nervous system. And so then we're going to have this sequence of events where we take the action potential in that neuron. We're going to pass it to the muscle cell that has this specific plasma membrane that we've given a new name, the sarcolemma. And that's going to cause our contraction. So this is going to be dependent on the fact that we have our neural input coming in down the nerve. We're releasing our neurotransmitter, our acetylcholine. And then that's going to cause the muscle fiber to release calcium from where it's being stored in the sarcoplasmic reticulum, in those lateral sacs that we looked at. So this is going to be kind of going back to our very brief look at neuromuscular junctions. This is basically that same concept, except, I mean, it's exactly that same concept really, except now we're going into a little more detail on what's happening in the muscle fiber in response to that signal. So steps, we're now kind of starting at the muscle fiber side, so from that neuromuscular junction. So from that somatic neuron, we had our action potential come to our axon terminal, but in a specific situation, it's called the terminal bouton. But it's just the same thing as an axon terminal in our normal neurons. Released acetylcholine, which was then taken in by the receptors on our sarcolemma on our muscle fiber side. This is going to cause an action potential, so depolarization, to travel across that membrane, across that muscle fiber membrane, which we call the sarcolemma. The action potential as it travels is going to hit the structure called T-tubules that we saw very briefly on Wednesday. So are these kind of tubes that go into the muscle fiber. So the action potential is going to travel through these T-tubules. There are receptors there in the T-tubules that open calcium channels in the lateral sacs. These are in parentheses because I don't need you to memorize those names. So just know that this action potential coming down the T-tubules is opening the calcium channels in the lateral sacs. That was that triad we kind of looked at in purple and green. As we open calcium channels, like we saw in our neurons in the central nervous system, when the calcium channels open, they're releasing calcium. In this case, they're releasing that calcium into the cytosol, so into the muscle cell. The calcium, once it's freely moving around in the cytosol, can come into contact with the filament. And when it bumps into troponin, our little regulator molecule that's sitting on our thin filaments on our actin, this shifts that tropomyosin, that thread that was hovering over our myosin binding sites on the actin, exposes them, and now we're able to enter a cross-bridge cycle because the binding sites are exposed, and this is a cell that is alive, so it can bind ATP. Do have energy to do this. So that's the whole kind of signaling pathway inside the muscle cell. We have a big map. Yeah, let's go through it one by one. So beginning here is our somatic neuron. So remember, somatic is the opposite of what when we're thinking divisions of the nervous system? Or any free word association with the word somatic. Okay, let's start out. Are we part of the central nervous system or the peripheral nervous system when we're talking about somatic nerves? Peripheral nerves, right? So we had, I'll do a brief review on the side while we're doing this. Okay, so we had our CNS, that was brain and spinal cord, sending out efferent nerves, motor nerves going out, and then everywhere we're talking about here, we're in the peripheral nervous system as we exit. CNS. Okay, so what are our two types of afferent nerves we might have coming out of the central nervous system? Somatic and autonomic. But that's helpful. Like automatic and autonomic are similar words because they kind of tell you what it means. So autonomic does kind of automatic functions. Somatic does voluntary active muscle contraction. Somatic versus autonomic. What we're talking about right now, here's the somatic nerves, but let's just make sure we have our whole split down. So what were our two types of autonomic nerves? Yep. Parasympathetic and sympathetic. What did the parasympathetic nerves do? Yep, both of them innervate the same effector organs because we have this dual innervation concept. So our effector organs are going to be cardiac muscle. Okay, so cardiac muscle, smooth muscle, and glands. So right now, we're way back here in our somatic nerves, which are separate from this whole process that we went through a long time and how we talked about how that integrates with our absorptive and post-absorptive states. Now we're over back to that little short, a brief teaser we did on the somatic nervous system. Okay. So this is our somatic nerve. So under voluntary control, so you've decided you want to contract, let's say your triceps, right? So your triceps muscle. So here we have our nerve coming in, goes to our axon terminal, which in this case, because we're talking about a neuromuscular junction, I label this the terminal bouton. Same thing as an axon terminal, but sometimes we use specialized terminology when we're talking about a neuromuscular junction. So just like with our other nerves that we saw in our last unit, we're then going to have that depolarization cause a change in the state of those voltage-gated calcium channels, which would tell these synaptic vesicles to release their neurotransmitter. And the neurotransmitter that is released by the somatic nervous system is always acetylcholine. So acetylcholine is the neurotransmitter for a somatic nerve. And the acetylcholine is the neurotransmitter that we will expect to see in the neuromuscular junction when we're doing these voluntary skeletal muscle contractions. So once we have the acetylcholine in this location, right, crossing now onto what we call the motor end plate. So the motor end plate is this twisty part of the sarcolemma, right? So this twisty membrane with lots and lots of cholinergic receptors here, right? So you have lots of receptors ready to receive that acetylcholine. So when these cholinergic receptors bind the acetylcholine, this causes what we call an end plate potential, an EPP. EPP is a depolarization. It means, would this be excitatory or inhibitory? It's always excitatory. Actually, always big enough that it causes the muscle cell to contract. So we don't have to add up graded potentials like we did for neurons. It's just when we get the signal, we're going to contract. Right. So those are kind of the steps that we saw when we looked at that one slide on the steps of the neuromuscular junction. Now we're thinking about what's happening in the muscle fiber. So our action potential is moving across the membrane. So as we start to see this depolarization happen, the depolarization is spreading out from the site where we had that contact between the nerve and the muscle cell. So this depolarization is happening in a wave traveling across the muscle fiber. So then it's going to come into contact with our T tubule. This is our T tubule. And it's diving into the muscle cell. It's diving into the muscle fiber where it comes into contact with the sarcoplasmic reticulum. This is the thing we referred to as our triad before. The triad was our T tubule plus two lateral sacs, so plus the sarcoplasmic reticulum on the side. The sarcoplasmic reticulum's job is mainly to hold calcium. So these little green things in here, That's our stored calcium. The muscle cell needs that calcium stored in there so that when it gets the signal, it can release the calcium in order to tell the muscle cell to contract. So as the action potential is moving down the T tubule and coming into contact with that sarcoplasmic reticulum, we're triggering the release of calcium. So calcium is going, where is it going now? It's staying in the cell, or is it going out of the cell? It's going to stay in the cell, so it's just going to the cytosol. So when we say it's releasing from the sarcoplasmic reticulum, we don't mean it's leaving the cell. We just mean it's leaving that organelle structure and getting into the rest of the muscle fiber so that it can come into contact with our filaments. So here we see the calcium binding into troponin. Troponin is this molecule that lets us push the tropomyosin aside. Right, so it's not very visible. Right, so here is our tropomyosin, and here is our troponin. When calcium binds onto the troponin, that pushes tropomyosin off the myosin binding sites. This is basically where we can now enter the cross-bridge cycle because we've exposed those binding sites. So next, see, so the cross-bridge cycle is beginning. The muscle fiber contracts as we push those thin filaments, that actin, towards the center of the sarcomere. And so we're pushing it towards the center. The sarcomere is shortening. Our A band, where the myosin is staying the same size, but our I band and our H zone are shortening. So those are about our regions of overlap or non-overlap. And as we turn off the signal, calcium can unbind, right? So calcium unbinds when we're done contracting. It gets transported back into the sarcoplasmic reticulum so that we can do this again. So we're going to hold on to that calcium so we can use it as a signal again next time you want to contract your muscle. So as we're unbinding that, right, so we've bound it, we're sending it back to the sarcoplasmic reticulum. We're then going to have the tropomyosin block the myosin binding sites again. It means our muscle fiber is going to relax as these basically slide back out, right? So the cross-bridge cycle kind of slid these filaments in towards the center and kind of held them there. And then when we cover off those binding sites, we just slowly have the release and the sarcomere lengthens. That's what relaxes our muscle fiber. So here we can zoom in on the shape that we see here. And so here we see the relaxed shape of those covered over binding sites. No calcium on the troponin. Here we see when the calcium is bound in, it's pulled the tropomyosin off of those myosin binding sites, so they're activated. In order to end a contraction, the calcium does have to leave the troponin. So as long as the calcium is present there in the cell bound to troponin, we're going to keep having this contraction happen. We're going to keep having that cross-bridge cycle. We're going to keep those sarcomeres short and tight. So we do actually have to actively remove the calcium from the cytosol at the end of this process. So there's something called calcium ATPase in the sarcoplasmic reticulum. And its job is basically to soak up excess calcium. So at the end of this cycle, when we're done having this depolarization telling our muscle to contract, those basically kind of like receptors are going to be sucking back in the calcium into the sarcoplasmic reticulum so that we can use them again. Let's take a look. one of the things that's a little difficult is the three-dimensional nature of these things.
Typically, a single motor neuron rising in the brain or spinal cord conducts action potentials that travel to hundreds of skeletal muscle fibers within a muscle. The sequence of events that converts action potentials in a muscle fiber to a contraction is known as excitation-contraction coupling. If we look at a single muscle fiber, we see that an action potential travels across the entire sarcolemma and is rapidly conducted into the interior of the muscle fiber by structures called transverse tubules. Transverse, or T-tubules, are regularly spaced enfoldings of the sarcolemma that branch extensively throughout the muscle fiber. At numerous junctions, the T-tubules make contact with a calcium-storing membranous network known as a sarcoplasmic reticulum, or SR. Where it abuts the T-tubule, the SR forms sac-like bulges called terminal cisternae. One portion of a T-tubule plus two adjacent terminal cisternae is known as a triad. The membranes of the T-tubule and terminal cisternae are linked by a series of proteins that control calcium release. As an action potential travels down the T-tubule, it causes a voltage-sensitive protein to change shape. This shape change opens a calcium release channel in the SR, allowing calcium ions to flood the sarcoplasm. This rapid influx of calcium triggers a contraction of the skeletal muscle fiber. Thus, calcium ions are responsible for the coupling of excitation to the contraction of skeletal muscle fibers.
So theoretically simple if we hadn't been doing so many other things already this unit. So, let's see. You know what? I actually want us to end here for today. Is there anything anybody wants to go over before we break? I will put up a quiz for this weekend, and I also heard that the practice quiz in studying for the exam was super helpful. So I'll make sure that we have one of those so you can do over and over again for studying. Any burning questions or requests at this moment?